All right, so the protein part was pretty straightforward. The DNA, RNA part is a little bit more involved only because there's so many things that we can do with DNA. So we'll do a few, and then we'll take a break, and then we'll wind it up. Every time I move the turning point thing, it comes out of slideshow mode, and it goes boop at me. Very annoying. Very annoying. Okay, recombinant DNA technology I'm not going to talk about. I'm going to assume that most of you know what this is, so I'll just spend 5 minutes on it, and, you know, if you have questions, you can talk to me at another time, or you can use the book or any biotechnology resource. Recombinant DNA technology means you're taking DNA from two different sources and recombining them to make a recombinant molecule. The DNA from the two different sources are: one is the gene that you want to study, which we refer to as your gene of interest. And you can be studying it for any variety of reasons, to just understand the gene or the way it is expressed or regulated, or you're just using the gene because you want to make the protein and study the protein, or use the protein for diagnostics or therapeutics or anything like that. Or maybe you want the gene itself to make genetically modified foods or genetically modified organisms that can be used for environmental bioremediation, things like that. So there's any number of reasons why you would want to be able to take your gene of interest and introduce it into a molecule, an organism that grows quickly and cheaply, so that you can have an inexhaustible supply of your gene of interest, and as a result of that, the protein product that the gene encodes. So the recombinant DNA technology is based on the understanding that there are enzymes known as restriction enzymes, which have the ability to restrict or cut DNA from any source as long as the restriction enzymes see their consensus or target sequence. So restriction enzymes have the ability to act as what's known as an endonuclease, endo for the inside. Okay, exonucleases chew in from the ends of a DNA molecule. Exonucleases cut inside. Endonucleases cut inside. Endonucleases are sequence specific, but they are not specific to a particular organism. If a restriction enzyme sees its recognition sequence or its target sequence, which is typically 6 nucleotides, it will cut. And it doesn't matter if the DNA comes from a bacteria or an elephant or a daffodil or whatever. It is just looking at the DNA sequence. So restriction enzymes cut DNA sequences in characteristic ways that can be recombined. So if you take DNA from two different sources and cut them with the same restriction enzyme, and then mix them together in the presence of ligase, which makes phosphodiester bonds, you can make a recombinant DNA molecule. Obviously, I'm skipping a lot of the nuances here, but I'm assuming that most of you have already learned this. So recombinant DNA molecules contain DNA from two different sources that have been recombined by using restriction enzymes to cut the two DNAs and ligase to seal the cut ends back together. So the way you get your gene of interest to be taken up by something that grows cheaply and quickly, like a bacteria, you're not going to introduce your gene of interest into the bacterial genome. That's far too complicated to deal with. Instead, you introduce your gene of interest into a small, autonomously replicating circular molecule that exists in bacteria as extra chromosomal. In other words, it's not the bacterial genome that's 4 million bases. It's a small, autonomously replicating circular DNA that's called a plasmid. These are naturally occurring in bacteria. They are how they share genetic information with one another, and scientists have co-opted plasmids and use them what's known as a cloning vector. So your gene of interest is actually part of a recombinant plasmid that has been taken up and is being maintained by the bacteria. So here's the bacteria. This is the E. coli genomic DNA, and this is the plasmid, obviously not drawn to scale, because the plasmid is like 5,000 base pairs, and the genome is like 4 million. Okay? So the plasmid has your gene of interest, and it is being maintained by the bacteria because you're using an antibiotic to select for the maintenance of the plasmid. And then you have these bacteria containing the plasmids that contain your gene of interest, and you can have an inexhaustible supply of the DNA. And if you use a plasmid that allows your gene of interest to be expressed, in other words, transcribed and translated by the bacterial machinery, then you can get the protein as well. So that's recombinant DNA in a nutshell. So let's talk a little bit more about these kinds of recombinant DNAs. You can make, like in that one I just showed you, making one recombinant plasmid that has one gene of interest. But what if you want to have the entire human genome represented in small genomic fragments inside plasmids? That's what's referred to as a library. Like a library is a selection of books where you could go and take out the book you want, although nobody does that anymore. That's what a library is. You can have a genomic library where all of the DNA fragments that exist in a genome have each been cloned or are now represented as recombinant DNA molecules. You can have genomic libraries where you cut the entire genome with restriction enzymes and clone each fragment and maintain them all as a collection. Or what many people do instead is they just make what's called a cDNA library. The C here stands for complementary, not DNA that you can see. Complementary DNA library, which means you take the messenger RNAs in a cell. Those are all the genes that are being expressed, and you use reverse transcriptase, a viral enzyme, which turns messenger RNA into double-stranded DNA, and then you clone that. So cDNA libraries contain only the sequences that were expressed in that cell. Genomic libraries contain the entire genome. So a human genomic library is going to be the same if the source was liver or brain. A cDNA library would be different because liver and brain are expressing different genes. So their messenger RNAs would be different. So the cDNAs would be different. So people use cDNA libraries to study patterns of gene expression. And they use genomic libraries to study genome structure, organization, mutations, things like that. Yeah? Oh, it's messenger RNA, so it's already been, yeah, in fact, they use the poly-A tail as a way to pull out the messenger RNA by binding it to something that's rich in Ts. Good question. We have our recombinant molecules, and we want to find a way to find the gene that we're interested in, our gene of interest. And so how do we do that? You guys already know, I'm sure. You take advantage of the property that all DNA molecules or nucleic acid molecules have, which is they have the ability to base pair with one another, specifically if their sequences are complementary to one another. So you have a DNA double helix. You can break the hydrogen bonds that hold the strands together by pH or temperature, right? Hydrogen bonds, you heat them up enough, they break. And then the strands separate or denature from one another. But then if you return the situation to normal pH, normal temperature, then the strands will renature or reanneal or reform the hydrogen bonds. So what that means is that you can take DNA from two different sources and denature them and combine them, and they will form a hybrid if their sequences are complementary to one another. So any two DNA fragments that are complementary to one another can base pair with one another, even if they come from different sources. So DNA from me and DNA from a frog, if we had the same sequence, which we might in some conserved genes, could hybridize or reanneal and form a hybrid double-stranded DNA molecule, one strand from me and one strand from the frog, as long as they can base pair. So hybridization means finding a DNA sequence using a molecule that can hybridize with it or base pair with it because its sequence is complementary. So the DNA molecules that you use to find the complementary sequences in your library are called probes. And they're probes because they are complementary to your sequence of interest, and they have some means that allow you to find them. They are radioactive or fluorescent or colorimetric or something like that. So you introduce a probe, it finds its complementary sequence and sits down on it, okay, hybridizes, base pairs with it, and then it announces its presence because it's radioactive or fluorescent or colored or whatever. So for example, if you take all of your DNA fragments from your libraries or anything else, you can separate the DNA fragments as a function of size by using a gel. The DNA gels are a little bit different from the protein gels, but I'm not going to take the time to elaborate on that. You can then allow the DNA fragments to leave the gel and immobilize them on something permanent and solid, a membrane or a blot. So you basically get an image of the way the DNA fragments were in your squishy gel, but now they're on some permanent, stable membrane or filter or blot. And then you can use a probe to find your DNA fragment because the probe can only hybridize to the fragment that it's complementary to. So this is referred to as a Southern blot, named after Ed Southern, who is the one who developed this technique of separating DNA fragments as a function of size on a gel, moving them out of the gel, basically wicking them out of the gel onto a solid membrane, which in those times was made of nitrocellulose and now made of nylon or something similar. And then you have a permanent record of all of the DNA molecules. The DNA molecules are actually there, fixed onto, baked pretty much onto the blot. And then if you treat them or incubate them with a particular probe, a short DNA fragment, it can be as short as 15 to 20 nucleotides, that is complementary to your sequence of interest, and in this case, radioactive. Your probes, the probe fragments will wash over all of the DNA fragments on the blot, but only stick where they are complementary. Then you wash off the unbound probe and put your blot over a piece of film, and the radioactive probe will expose the film, and you'll see where your fragments are on your gel. So this is called a Southern blot because of Ed Southern, and this was a DNA gel followed by hybridization. Since then, there's Northern blots, as I'm sure most of you already know. Northern blots, not named after somebody called Northern, but just because it's opposite of South. Northern blots are RNA, and Western blots are doing the same thing, more or less, with protein. Although in Western blots you're not using hybridization because proteins don't have complementary sequences. In Western blots your proteins are serving as antigens and you're using antibodies that are specific to your proteins. So Southerns, Northerns, and Westerns are the same in that you're taking a huge complex population of DNA or RNA or protein, resolving it through a gel, transferring it onto some permanent, solid, manageable matrix, and then using some sort of probe to specifically track your DNA, RNA, or protein of interest. Okay, PCR. Again, my 311 people are going to recognize this. So PCR is one of the most revolutionary technologies to come along. Came along when I was just about done with my postdoc, which was unfortunate because it would have made my postdoctoral experience so much more pleasant if PCR was around. But anyway, PCR stands for polymerase chain reaction. It's an in vitro DNA replication, but you're not synthesizing or replicating the entire genome. You're replicating a specific defined sequence, which you are defining by the way you set up the PCR reaction. So DNA synthesis in vitro requires a template, right? Double-stranded DNA. Requires polymerase, of course, nucleotides, and it requires a 3' hydroxyl that DNA polymerase can elongate from. So to do PCR or DNA synthesis in vitro, the primer is not provided by the primase enzyme. The primer is provided by the investigator. So to do in vitro synthesis, you have your DNA template, which is your genomic DNA or your clone DNA, whatever you're using, and then you provide the primers. These are 2 short, say 15 minimum nucleotides long. Your primers anneal to opposite strands of the template with their 3' ends pointing towards one another, okay, like these guys in blue, okay? And then, so the PCR is a combination of, is a reiteration of 3 steps. The template DNA is denatured, so in real life it's denatured by helicase enzymes. In PCR, because you're in a test tube, it's denatured by heat. Then you bring the temperature a little bit down to allow the primers to anneal, because if the temperature is too hot, the primers can't base pair with the template because hydrogen bonds are not stable in the presence of high heat. So you bring the temperature down to allow the primers to anneal, and then you extend. There's a polymerase there that can extend or synthesize DNA. So all of the cycles are very short, say 30 seconds to denature, 30 seconds to anneal, 30 seconds to extend. The polymerase used in PCR can do at least 1 kilobase a minute. So typically the size of the fragment that people are amplifying is a couple of hundred to a couple of thousand nucleotides. So a 1-minute extension or a 1.5-minute extension is usually fine. You can do 30 cycles in an hour and a half or so, and at each cycle the amount of target DNA doubles because you're synthesizing 1 copy at each cycle. So you can have 1 million copies of your target DNA, the DNA in between the 2 primers, in a morning. So it's truly revolutionized by removing one of the major obstacles that researchers always had, is that they had a limited amount of DNA to work with, especially people who are looking at DNA like from forensic crime scenes or DNA from tumor biopsies or DNA from prenatal diagnostic samples or DNA from archaeological digs, you know, you're limited in the amount of sample that you have. So you're limited in the number of things you can do, especially if you want to reproduce your experiments. So having PCR allows you to amplify and get a million billion-fold increase in the number of copies of your DNA, and then that removes the whole anxiety that comes along with having a limited amount of sample to work with. So again, you have your template, which can be genomic DNA or clone or a sample. You denature the two strands at a high temperature. You bring the temperature down a little bit. You allow the primers to anneal. You adjust the sequence of the primers and the annealing temperature so that the primers only anneal at sequences they are perfectly complementary to and not at 17 other places in the DNA, which would mean you're amplifying more than what you think you're amplifying. And then the extension allows the primers to be extended, as you can see here, synthesizing new DNA based on, you know, being complementary to the template. PCR, now that it has been automated, uses a polymerase enzyme that comes not from us or bacteria, but instead from an organism that lives at a high temperature. So the polymerase can polymerize at an elevated temperature, 72 degrees. So it's a thermostable polymerase, comes from an organism abbreviated TAC, stands for Thermophilus aquatis, something like that. Hot water comes from the hot springs. And that's good because then you can keep the temperature nice and high. That means that you never have to bring the temperature down so low where your primers can anneal non-specifically. And also it means that this thermostable polymerase is a protein, doesn't itself denature or become inactive when you raise the temperature up to 95 to denature the DNA. So you can denature the DNA in the presence of the polymerase enzyme, and it is not harmed by that elevated temperature. So the identification of the thermostable polymerase allowed PCR to be automated. Now there are PCR machines or thermocyclers, which are the size of a shoebox, and not like the original ones, which were huge. Everybody can have one on their bench top, and 1, 2, 3, you're good to go. Okay, so we can use PCR to amplify any DNA. The DNA can come from the genome directly. It can come from libraries, and it can also be, you can use it to characterize cDNAs, because once you have generated cDNA from messenger RNA, you can amplify the cDNA that you're interested in by using primers that are specific to that sequence. Okay. So we'll come back to PCR a little bit when we talk about mutagenesis and characterizing gene expression. So sequencing, everybody knows that sequencing has been completely automated and gone through several rounds of improving the automation and robotic nature of it. When I was a postdoc, it took 2 years to sequence a 3 kilobase intron. Now it can be done overnight. Everything has completely changed in the speed and the automation of it all, so that, you know, sequencing of genomes has become, I wouldn't say trivial, but, you know, nowhere near the kind of, you know, galactic effort that it was when the human genome was first sequenced almost 20 years ago. Okay. So the original way of doing sequencing, well, not the original, but the most common way of doing sequencing up until recently, and is still done in many labs, is sequencing with what's called dideoxy chain terminators. So to synthesize DNA, you need a 3' hydroxyl, right, as you know. So if deoxynucleotides are missing the 3' hydroxyl as well as the 2' hydroxyl, they're called dideoxy, di for 2. So deoxys are just missing the hydroxyl on carbon 2. Dideoxy are missing the hydroxyl on 2 and 3. So if you incorporate one of these into a DNA molecule that terminates the synthesis, because there's no 3' hydroxyl, that's why they're called chain terminators. So if you want to read the sequence of a DNA molecule, you can synthesize it in vitro in the presence of template and primer and lots of normal DNTPs to get the synthesis going, and trace amounts of dideoxys, which will randomly be incorporated, and when they are, they will terminate the chain. So you will get a whole collection of DNA molecules of varying lengths, from 5 to 10 nucleotides long to 1,000 nucleotides long. Each one will end with a chain terminator. So if you include your dideoxy chain terminators, and each of them is fluorescently labeled with a different color, so A is one color, G is another color, T is a third color, and C is a fourth color, you'll generate a whole array of fragments. Each one will be a different size, and each one will fluoresce in the color of its terminal nucleotide. So if you have a fragment that's 17 nucleotides long and it's fluorescing in the color of A, the nucleotide number 17 is an A. Okay, so you take this collection of synthesis molecules and you separate them on something that separates as a function of size, but it has very, very high resolution such that even a 1 nucleotide difference can be distinguished. So it's electrophoresis through like a capillary tube. And then of course, it's seen by something that is reading the fluorescence of the nucleotides as they are coming out the bottom end. They're running through as a function of size. The smallest ones come out first, the largest ones come out last. So it's all automated, and it's reading the color of each band that comes off and converting it into something that looks like this. Is a chromatogram, and you can just look at it and see how nicely you can see each nucleotide one after another. Okay. So since then, there's been second generation and even third generation sequencing. So much improvement in the technology and the automation that it cost $100 million to sequence the human genome by 2001. Now it costs not even $1,000 to sequence a genome, and before too long, getting your genome sequence will be like determining your blood type. It'll be a couple of $100, and it can be done very simply. And then, of course, the question is, what does the information mean, and whose has it, and who has access to it, and all of that. But that's, a problem that, was not something that could have been anticipated 10 years ago, when it took a huge amount of effort and time and money to sequence the first human genome that was sequenced. So now there's next gen and even third generation sequencing. I'm not going to go through them in detail, but just to give you an idea of the power of this kind of sequencing. In this kind of sequencing, you're not sequencing one fragment at a time. You're actually sequencing thousands of different fragments all at a time. You can take the DNA and fragment it and amplify individual fragments separately. You can attach them to beads and put them in these individual wells. So you have millions of different templates all being sequenced simultaneously. And again, of course, this is all done robotically. The way this kind of sequencing works is that you have a whole bunch of different templates. Each one is in a different well of a microtiter plate, and you add a particular nucleotide, say A, to each of these wells. If A was the next nucleotide and it gets incorporated, then of course the DNA polymerase adds the A in making that phosphodiester bond. The pyrophosphate is released. The machine that's doing the sequencing is going to detect the pyrophosphate release. So if an A was the next nucleotide in however many of these wells, they will give a signal that's detecting the pyrophosphate. Then, of course, C is added, and G is added, and T is added. So each well is going to fluoresce when one of the four, or signal when one of the four nucleotides is added. So all of this is done, you know, like I said, robotically, and what's being detected is the flash that comes when the nucleotide has been incorporated and the pyrophosphate is released. So this is all done. You're sequencing lots of different templates all at once. The machine is just offering the template, an A, no, a C, no, a G, flash. Okay, so G was the next nucleotide in that one. So you're sequencing a whole bunch of different templates all at the same time by detecting whether or not there's been a flash when a particular nucleotide has been added. So this is next generation sequencing, and there's even advances beyond that. So everything has become totally automated, and the amount of time and energy and brilliance that has gone into doing this is absolutely astounding to people of my generation when we think of how long it took us to sequence, you know, 600 base pairs. It was an achievement, and now, you know, in the time you get a cup of coffee, that can be done. And it's because young people like you who saw an opportunity to use their talent and their education to advance the field, you know, put the time and the effort in, and this is what happened. So it's amazing.